All right. So we've been doing this a long time, and in fact, I've been thinking it's probably 8,000 students have taken this, or something crazy number like that. It's wild. But this is for some of you, this is going to be scary, but just chill. I'm going to repeat things multiple times. The slides are posted. You can email me any questions you have. I'm also going to be back later in the semester, and we can talk some more. So just to give you I need a pointer. I do have a pointer? I do. No? Yes?
So if you want it to be a pointer, you would.
Okay.
And then you press this button.
Okay. Got it. All right, so just to give you a history of this, so this is my picture starting when I was a little baby, because feels like how long I've been working on this. I actually first saw one of the first patients with HIV in 1980 in New York City when I was an intern and then resident in the Bronx. But it was first officially described and recognized as a new syndrome in 81. The virus was isolated in 83, and by 85 we had the first HIV tests so they could test blood. So prior to that, a large number of people were getting infected both through the blood system that was contaminated from donors who didn't know they were infected. It was found to be transmitted from mother to child, from shared needles from intravenous drug users, and through sex, both heterosexual and homosexual sex. Do you know who this is? Liz Taylor just passed away. She was one of the incredible heroes. So a lot of actors talk about getting involved in things, but she walked the walk. This woman, she would fly to Palm Beach and in Florida, and she would literally go door to door knocking on mansions to raise money for HIV. In an era when everyone was running away from patients, they didn't want to touch them, they didn't want to have anything to do with it, and she just plunged right in as she saw friends become ill and die. And she really was heroic effort. This guy's Michael Gottlieb. He's the first guy to recognize in his practice that something bad was going on, and he called up the CDC. And these are the two guys who discovered the virus. Oops, wait, sorry. Okay, so a little more history, and then we're going to get into the difficult stuff. So first drug was shown, despite the epidemic going on, and everyone was thinking this was going to be the end of the world, an enormous amount of progress was made. So 86, AZT was shown to be effective. The first drug was discovered by Burroughs Wellcome Corporation. It was now part of GlaxoSmithKline. 86, the National Institute of Health funded the AIDS Clinical Trials Group, which started testing these drugs in patients, and an enormous amount of patients came forward to be in studies. 1990, Congress passed the Ryan White Act, CARE Act, which provides funds for treatment and medications. Then Congress passed in 92 housing opportunities for persons with AIDS who were homeless. In 94, we showed that we could actually stop transmission from mother to children, and now, prior to then, hundreds of babies were getting infected every year in the United States, and now, literally, throughout the whole United States, maybe 5 babies a year. In fact, there are more HIV pediatricians than there are babies for them to take care of. And then by 97, we figured out in a study that we did here and other centers that triple drug therapy could completely suppress the virus in the blood so that patients could potentially have a normal lifespan. And my oldest patient was infected in 1983, and he's still going strong. That is Trudy Ellian. She won the Nobel Prize, and that's me when I had a beard. And she had won the Nobel Prize. She lived here on Cameron Avenue, worked till the day she died. She was the co-discoverer with George Hitchings of many of the drugs we use to treat cancer, that we allow organs to not be rejected, and to treat all sorts of virus infection and parasite infections. She was a terrific lady. Okay, how bad is the problem? It's bad. Currently, as of 2007, there are up to 36 million people living with HIV. New people get infected every year, and the worst, of course, is in sub-Saharan Africa. In terms of the numbers, however, China is a big fear for people, as is India, because the populations are so big, even though the percentage of people infected is small. In the U.S., we continue to have about 60 to 70,000 new infections each year. Nothing's changed. So that's not good. We're not doing a good job there. Okay. And here are some of these numbers here. We see 22.4 million in sub-Saharan Africa are infected. Okay. So the natural history is you have sex here, and within 2 weeks, usually 2 to 4 weeks, most people, but not everybody, will get flu-like symptoms, a fever, sore throat. They might get a rash. They get a huge burst of virus in the blood. Okay? If you did the standard antibody test and someone came to the emergency room, would you be able to detect HIV antibodies then? Yes? No? No. Right. Very good. No, it takes a while for the antibodies to kick in. You don't detect them for usually 2 months, 8 weeks. So in the beginning, someone will you have to do a test to measure the virus itself to detect infection, okay? Sex here, virus gets in, huge burst of virus. This is upwards of 3 million copies of the virus per drop of blood, mil of blood. Eventually the immune system kicks in, antibodies are made, lymphocytes that are part of the white cells in the blood attack HIV, decrease the amount of virus in the blood, but it's still present, doesn't go away. And here the patient will feel sick. Once the immune system kicks in, the symptoms go away. Okay, and the patient is infected and infectious, so the virus can be transmitted, but they don't know it, okay? The antibody test is positive. The cell that gets infected by HIV is called the CD4 lymphocyte. It's a white cell in the blood. It remains, a normal level is above 500 cells per mil of blood. Over time, the virus wins, the immune system loses, the CD4 count drops, okay, and then the viral load will go up, and the patient becomes symptomatic, develops symptoms again and gets sick. So we talk about the HIV iceberg. We have a huge number of people where you don't know they're infected, okay. We have a smaller group, but still large, where there are minor little symptoms and infections. And then only at the tip do you get what's called AIDS, and that's a much smaller number. Okay, let's look at the virus, and we're going to break down some of the things I talked about into details, okay? So this is the virus. It is what's called its genetic material is 2 single strands of RNA. All right, what's our genetic material? DNA, very good. Double-stranded DNA. This is single-stranded RNA, okay? It has this capsule around it, all right? A core capsule that's around it. It also has 3 enzymes that are unique to the virus, okay? And those are important because that's the reason that Trudy Ellian won the Nobel Prize is she figured out the way we need to develop medications is to understand the way these viruses and parasites and fungi and cells work, and then we should target particular parts of it. So HIV has 3 enzymes. It has the reverse transcriptase enzyme, here colored blue, that actually makes a DNA copy of the viral genetic material. It has the integrase, which takes that double-stranded DNA, the viral DNA, brings it into the nucleus and sticks it into the patient's chromosomes, okay, of the CD4 cell. And then it has the protease enzyme that when viral proteins are made, it cuts them into nice little pieces into the individual proteins. The virus is surrounded by this lipid or fatty bilayer, bi meaning 2, okay, and which makes it easy to kill the virus using soap and alcohol and things like that, because those things dissolve fat. On the surface of the virus is a protein called GP120, which stands for glycoprotein 120. 120, it's its molecular weight. And this is the key that unlocks the door of the CD4 cell. It's how it gets into the CD4 cell. It's why not everyone in this room is infected with HIV. You can't cough it onto somebody because it has to have a specific receptor that this key can get into. Okay, close-up virus. You see, here's our lipid bilayer, GP120, glycoprotein 120 on the surface. This is the lymphocyte, the CD4 lymphocyte is sometimes called a T4 cell, used to be in the old days. So this is the CD4 protein. Those two stick to each other, doesn't stick to other proteins, only sticks to the CD4. All right? We subsequently found, and this was really brilliant work that was done by 2 different labs, that there were people who they had CD4 cells, but they were having unprotected sex with people who were HIV infected, but they weren't getting infected. So what was that? Okay, and what it was is because everyone thought all you needed to get infected with HIV is the virus and a CD4 cell, and you get infected. But these scientists they looked at the people who weren't getting infected, and they weren't getting infected because they lacked another receptor. So you actually need 2 receptors to get infected with HIV. You need the CD4 receptor here, the little green thing, and you need what's called a chemokine receptor, or CCR5, okay? You need both of those, and there are about 1% of Caucasians lack that receptor, so they can't get infected. All right? And you may have read about the person who was cured of HIV. Did you read about that? The Berlin patient. Some of you who are in sciences may have heard about. There was a guy who was cured, and what it was is he was HIV infected, and he had a bone marrow transplant, and the donor, they picked someone who lacked that receptor. So before you get a bone marrow transplant, you kill all the lymphocytes in a patient, all right? Then you give them new lymphocytes from a donor. So they killed all the lymphocytes in the patient, and then the donor lymphocytes lacked that receptor, and so he survived, lucky, his bone marrow transplant, and his malignancy didn't come back, and his HIV was gone. So anyways, the way it works is that it's really very cool, and if you get a chance, there's some YouTube videos, which if I was clever enough to know how to do YouTube into my talks, I would have done it, but I'm not that clever. And so what it is, it has a stop-motion picture of what actually happens. So you've got here's your CD4 cell, the cell membrane, CD4 protein. This is the virus. And sort of landing gear, there's the virus there. It has GP120, you see there. First it sticks to the CD4, then the CD4 protein draws it down to the chemokine receptor, and then in a very sexual looking kind of thing, the virus jams in a protein into the membrane, and the 2 membranes, the cell membrane and the viral membrane, fuse. No, it does. No, I mean, it does sort of sexual, yeah, right? Okay, so this is the virus. This is the CD4 cell, and so you see the membranes fuse, and the capsule of the virus enters there. Here's a big view of virus coming in, little spaceship here, the mothership. GP120 sticking to CD4 protein, membranes fuse, the capsule is inserted, the core protein there, you got your little naked RNA there, it's not ready. The reverse transcriptase then changes it from single-stranded RNA, it makes first a single DNA copy of it, and then another copy, making it double-stranded DNA. Then the integrase enzyme takes it into the nucleus, okay, and it glues it into the patient's chromosome. Then the host enzymes, your own cell enzymes take over, and copies of the viral RNA are made. Messenger RNA is made, it leaves the cell nucleus, proteins are made, and the final enzyme, the protease enzyme, then cuts that amino acid chain into pieces of individual proteins. Then the virus is packaged and it buds out, leaving the cell. So you can see there are multiple places that understanding this was important to designing the drugs necessary to fight this virus, okay? So now we have drugs that block the sticking of the GP120 and the CD4. We have multiple types of reverse transcriptase inhibitors. We now have, I think, 2 integrase inhibitors. We have multiple protease inhibitors, and they're actually designing drugs that will block the budding out of the virus. Okay? Just Watson and Crick, who discovered double-stranded DNA. Just a little history there. So you're not going to obviously know anything about this, nor will you ever be tested on this, but this is the genetic structure of HIV. It has these main proteins that we know about, that you know about too. So the POL gene, POL, standing for polymerase, it makes those 3 enzymes. It makes the reverse transcriptase and the integrase and the protease, all right? It has the envelope gene, okay, that makes the CD4, the GP120, okay? And then those core, that little capsule, the space capsule looking thing, that's made by GAG. And scientists are busy trying to find out what all these other things do, and then design new drugs will target those other things. There's a picture of a CD4 cell being invaded by a bunch of virions. Here's an electron micrograph. Here's the CD4 cell. These are the viruses budding out of it. This is a man with acute HIV infection. That's his eyebrow, hair there, forehead. You can see that faint rash, red rash. This just looks identical to infectious mononucleosis, okay? And this is an aphthous ulcer, teeth, tongue. Aphthous ulcer, that is very characteristic. If someone comes in with mononucleosis and has an aphthous ulcer, think HIV. These are the symptoms you get with acute HIV. You get fever, feeling fatigued or tired, sore throat, weight loss. You can get myalgias, which means muscle aching, and headache. About 50 to 70% of patients will get that. How many am I making neurotic? None of you are going to have sex for at least 6 weeks. I'm sure of that. Okay, so once you get the infection, it goes, so years ago when I got this really cute letter from a middle schooler in North Carolina, because he came up with, I thought it was a brilliant idea. It said, since it infects cells in the blood, why don't you just take all the blood out and heat it up and kill the virus? Not a bad idea for a middle schooler. So, and the problem is, of course, is that the virus goes throughout where immune cells go. And immune cells, we now know, go everywhere. They go to the brain, so they're brain macrophages and brain glial cells. They go into the lymph nodes all over. They go into the gut, the colon, the small intestine, etc. So they're all in the bone marrow, so it's all over. It's not just in the blood. Okay, that was the virus. Now we're going into immunology, basic, and those of you who know something about this realize I know nothing about this. So I'm just sort of giving a general overview of the immune system. So what protects us from disease? We're protected by our skin, our mucous membranes, by humoral or antibody-mediated immunity that's made by B cells, and cell-mediated immunity, other lymphocytes. This is a slide of a micrograph of skin, and you can see that you've got this. The reason why we don't get infections is because on the layer of our skin there are no live cells. Okay, that's just a thick layer that's oily, and things can't get through it, and that's why we don't get infections. Whereas if you cut yourself, bacteria can get into this layer, the cell layer, and it will lead to an infection. Okay, so let's look at those. So all these cells start out with a stem cell, okay? So it's a single cell from which all the other immune cells come to be. So we have these cells become the T cells. For instance, they go to the thymus to get educated. You got the CD4 cell there, sometimes called the T4. That's the cell that gets infected with HIV. We've got the CD8 cell. That's the cell that will kill cells that are infected with viruses. And then we have natural killer cells. I'm not going to go into great detail on all those. We also have B cells that are made that come from the stem cell. Those make antibodies, okay? And then we have other cells that are made. So what's an antigen? We're going to get into this. I'm going to put it all together. We've got little pieces, and then we're going to put it in the big picture. So hang in there. An antigen is a molecule which is recognized by an antibody and by cells of the immune system. So if you have a virus, and you saw how that virus had all those different parts to it. It had proteins, it had RNA, etc. If you break up those proteins into little pieces, okay, or the glycoproteins, etc., those are antigens. That's what antibodies will recognize, okay? All right, this is Immunology 101. It shows the whole system, so let's walk through it. So you've got, suppose you're exposed to herpes simplex, okay? What first happens is that one of the cells in your body recognizes this as a foreign invader, okay? This herpes doesn't belong here, all right? So it then chews it up. It's called the macrophage or antigen-presenting cell. It chews it up, and it puts pieces of that foreign invader, of the herpes, on the surface of the cell. Those are antigens, okay? And then what it does is it shows it to the rest of the immune system. Remember the old prison breakout movies where they had the bloodhound and they got the t-shirt or the underwear of the escaped prisoner? So that's what this is. So this is the antigen is the stinky undershirt that they're given to the dog, and the dog is going to go hunting down the thing. So little pieces of herpes simplex are on the surface of the macrophage. The macrophage then goes and it starts sweet talking to the CD4 cell, and it says, look, we're being invaded by herpes simplex. You need to get off your ass and go after the herpes, okay? So the CD4 cell does two things. It increases a number of cells that are specific for that particular antigen, okay? So it will go after one particular piece. So this CD4 is against one part of the herpes simplex. Another CD4 will be against another part of herpes simplex, okay? Oops, shoot. Then what happens is that the CD4 cell not only increases in number specific to the individual antigens or pieces of the herpes simplex, it starts talking to the B cells that they should start making antibodies against herpes simplex, that particular antigen, and the CD8 cells, the killer cells, the cells that are going to go out and kill cells that are infected with herpes simplex. So in the end then, what happens, you get all these different B cells that are making antibodies that will stick to the herpes simplex. You got all these CD8 cells that will go ahead and kill cells that are infected with herpes simplex. So you're coming against the herpes from two ways. Herpes that's floating around free outside of the cell will be killed by the antibodies. Herpes that is in a cell but has pieces of the herpes on the surface, those cells will be killed, and you wipe out the herpes simplex, okay, if you're successful. All right, and then after the infection is cleared, you're left with memory cells that remember that infection. You remember your memory B cells, memory CD4 cells, and memory CD8 cells. And they're there for years and years and years, and that if you get exposed to herpes simplex again, you can kill it. The reality is herpes simplex is actually a bad choice because herpes simplex is very hard to eradicate. Most people don't eradicate. Hepatitis B would be a better example, where most people can clear hepatitis B when the immune system gets in. The reason I said herpes simplex is because a new vaccine study that was done partly here has shown that it does work. Anyways, a vaccine, what it does is it takes part of hepatitis B. It takes an antigen, and instead of giving you the live virus, it just gives you the antigen. But the same thing happens. The macrophages chew up that antigen, put it on the surface, it turns on the CD4 cells, turns on the B cells, turns on the CD8 cells without any infection, but you're left with memory cells against hepatitis B. Then when you're exposed to the actual virus, you have sex with somebody who has hepatitis B, all those memory cells come alive. They've been sleeping, like some of you are now. They've been sleeping, they wake up, they increase in number, and they fight that in the hepatitis B so that you don't get infected. That's how a vaccine works. Now, what's the big problem with HIV? What does HIV infect? Louder. CD4 cells, okay? So HIV is infecting the very thing that is crucial to control HIV, right? So it's a diabolical virus, and you can see how some people in the early days thought it was like a man-made kind of conspiracy theory. Because what it does is HIV, normally you get infected with HIV, and macrophages would chew it up. It would turn on CD4, CD8, okay, B cells to make antibodies. But what happens? The CD8 cells are going to kill what? The CD4 cells that are infected with HIV. So eventually, the virus wins, the CD4 count drops down, and you lose all your immune system, okay? So that means that all the vaccines, that you're immune to, they're going to stop working eventually, okay? And we also, we'll go now. All right, so that's the general overview. We're going to go quickly through these, and then I'll come back to this. Okay. So here's a breakdown of it. We got the macrophage. There's the nasty virus. Chews it up. Puts a piece of the virus or the antigen on the surface. Here's another picture. Here's the macrophage. There's the antigen. It's presenting the antigen to the CD4 cell. It's like they're having sex. They're talking, and they're saying, this is a bad thing. So the macrophage is telling the CD4 cell, this is a bad thing. You need to do something about it. Okay? And remember, we have the exposed, the antigen turns on the CD4, you get your immune response, all right? But then you have memory cells. If you get exposed to the same antigen or virus again, you get a massive response, like with a vaccine. Okay. And as I told you, there's two parts of the antibody-mediated response, which the B cells will make antibodies, and you get the cell-mediated response where you get these memory and cytotoxic lymphocytes that target cells with antigen on their surface, okay? Direct killing of infected cells. And here's to show those cartoons aren't just made up, they're real life. You got a T cell, a B cell, a macrophage all talking to each other. This is what an antibody looks like, okay? The antigen sticks in there in that Y shape. This shows you a virus with various antibodies sticking to it. That's a real live one. And here you've got the CD8 cell that will, you've got the CD4 cell that's infected with HIV, and on the outside of the CD4 cell, you've got a little piece of the HIV, the antigen there. The CD8 cell sees it and it kills it, okay? You end up with a dead CD4 cells, and the antibodies against HIV, they go after cell-free virus. Okay, I don't think I need to go through this. You know, but so you end up then, you got HIV infects the cell, you have the single-stranded RNA. First, one DNA strand copy is made, then the double-stranded DNA, that's done by the reverse transcriptase, and then how does it get into the nucleus and become integrated in there? Integrase, very good. And then the proteins are made, okay, and the protease enzyme cleaves it, and it buds out, and it can end up killing the CD4 cell. So you can lose CD4 cells two ways. Either the virus can kill the CD4 cell itself, or the CD8 cells can target the CD4 cell and kill it. Okay. So we know that the virus in the body is in various places. So there is free virus, okay, floating around in the blood in a patient who's not on therapy. That has a very short half-life, only about 6 hours, okay? Then that will infect the CD4 cells, and those will be is a larger pool. It's 99% of the virus in the blood. It's in the CD4 cells. It will release new virus, and then the cycle goes on unless you treat the patient. There is also what we call latently infected CD4 cells. So CD4 cells that have virus, but they're not dying. Okay, they're not producing much virus, they're just sitting there latent. And that's a problem, because when you start treating somebody and interrupt the reverse transcriptase, the protease, the integrase, this will disappear in the blood within 2 weeks. So I can have somebody with 2 million copies of virus in their blood, and if I start them on 3-drug therapy, this pool will be gone in 2 weeks. This pool will be gone in 3 months, okay? And you can treat somebody we've been doing it now for years and years and years, and that you cannot measure the virus there. It can be on standard tests, okay? It's completely gone, except in these latently infected cells, okay? It's still there. And that's why we haven't cured anybody using standard methods. But we've got a huge group here working on campus led by David Margolis and Victor Garcia looking for ways to do that. Okay, one of the things we do is we measure the do you all know what a polymerase chain reaction is, PCR? How many don't know? Raise your hand. Ooh, a lot of people don't. Very important you know it, because it's going to be used on you at some point, okay, in your life. So this was, another person got the Nobel Prize for this. So the big issue is, how do you detect things that are in very, very small quantities in your body, okay? In the old days, you know, to measure HIV, we'd have to take the blood and we'd have to put it on cultures of CD4 lymphocytes, all right? That's dangerous to the person drawing the blood, culturing the cells, etc. Very expensive. You need special hoods to do it in. So we really couldn't do that except in research settings. But then a guy came along and discovered this clever idea. So what it is, you have whatever genetic material you want to measure, RNA or DNA, and you heat it up. In this case, it's DNA, double-stranded DNA. If you warm it up a little bit, the two strands come uncoiled, okay? You then put in what's called a probe, all right? And these little pieces are that will stick to the DNA on either side of the thing you want to detect. So in this case, we want to measure the HIV that's in the DNA of the patient, okay? So we take the patient's blood, separate out the DNA, okay? We then put little probes that are copies of HIV, but little pieces of it, not the whole long genetic material, and it sticks to the patient's chromosome where it has the virus in there, okay? And then we make a copy. We put an enzyme to make a copy of it, just like the equivalent of the reverse transcriptase, okay? So it then makes a little copy, all right? So you end up with, you have your original copy of your virus here in the orange and red, and then you have two more copies, okay? So you've doubled it, all right? You then heat it up, those strands come apart, you've got the probe in there, it now sticks to 4 strands, all right? And you make copies, and all of a sudden you double it again. And you keep doubling and doubling, so you repeat, you get a heat, copy, cool, heat, copy, cool, and soon you end up with 1,000 amplifications of that little thing that you couldn't detect before. And that's how, and you can do it safely, because this is just DNA. It's not a cell, it's not a whole virus that can infect someone, okay? So that was the discovery, and that's how we now monitor patients. We can tell a patient how much virus they have in their blood when we see them. So remember, you have sex here, you have your huge burst of virus when you get infected. If you try to do an HIV antibody test, it's negative then because they haven't developed antibodies. Then the immune system kicks in, you have your cellular immunity, your CD8 cells drive down the amount of virus by killing cells that are infected with HIV. Antibodies are made that drive down free virus, so once the immune system kicks in, the amount of virus goes down in the blood to a particular threshold that stays the same forever, okay? What determines why someone has only 1,000 copies of virus in the blood per mil versus 100,000, okay? It's a very important question, because if you have 100,000 copies of virus in your blood, you're going to develop AIDS and get sick much, much faster than if you have only 1,000 or 10,000 copies. What's the difference? It's how many antigens your immune system is targeting. So for some people, and we don't know why this is yet, for some people, the immune system response to HIV and other things is, it may be, and suppose I'm HIV, it may be that I would only have an antibody to my right 5th finger, okay, or my nose, all right. But for other people, they're covered. A bunch of antigens are targeted on the virus, okay, all over the place. And that's those people with a more robust immune response, okay, where the CD4 cells and the B cells and the CD8 cells are recognizing many more pieces of HIV, those folks have very low virus in the blood, and they're quite lucky. And it's very important. So if you have, these were patients who were not treated and what their prognosis was, a very old paper done by a friend of mine. So the viral load in someone who had less than 3 copies per milliliter, in 9 years, only 11% had developed AIDS. Whereas if they had over 100,000, 76% had developed AIDS. And so how do I explain it to my patients? I tell them, I say, well, this is the way it is. We're all on a train heading for a cliff, okay? We're going to go off someday. The CD4 count is how much track you have, okay? If you have a CD4 count of 500 or 600, you got a long track, a long way to go. You can't see the canyon, okay? If you have a CD4 count of 10, you're standing at the edge of the Grand Canyon. The viral load is how fast the engine's going down the track, okay? If you've got a viral load of 100,000 copies, it's going lickety-split, all right? So obviously the ideal way for your newly diagnosed with HIV is that you have a CD4 count of 500 and a viral load of 3,000 copies, okay? Then you got a lot of track, and your rate of progression, your rate of loss of CD4 cells is very slow. Normally you start above 500 CD4 cells, okay? You have your acute infection, that's when you have symptoms. Then that slowly decreases over time. On average, about 50 cells a year. So, you know, on average, it'll be 10 years before bad things happen. But it's highly variable depending on the viral load, as I said. The definition of AIDS is less than 200 cells. We talked about this already. As your CD4 count drops down, you start developing serious infections. First minor ones and then serious. Now most of you are under the delusion that you're paragons of virtue. But in the reality is, you're all cesspools of vermin. What I mean is that when you were children, you became infected with a lot of different viruses and parasites, only you didn't know about it, okay? And the reason you don't know about it is that you have intact CD4 cells, CD8 cells, and B cells that keep those latent infections latent. They're quiet, okay? So some examples. About 80 to 90% of you are infected with CMV, or cytomegalovirus, okay? It's one of the herpes viruses, and 90% of you are infected. It's latent in your white cells, and you'll never know you have it until you get a kidney transplant and we have to knock down your immune system, it'll start growing, or a heart transplant or lung transplant, until you get HIV and your CD4 count drops below 25, or you get a bad malignancy and we have to give you chemotherapy and knock down your lymphocytes, okay? Then it'll start growing. And that's true with most of the infections that people with HIV get. So you're not when you get someone who's seriously sick with HIV, with some weird infections, most of you are immune to those infections already and you're not going to get them. So tuberculosis, when the HIV patients get it, it's usually reactivated disease. They got it from grandpa when they were kids. It's latent in their bodies and it reactivates. Shingles is due to the chickenpox virus, also called varicella-zoster virus, and it's latent in the nerve endings. When the immune system starts deteriorating, it can reactivate. Herpes will reactivate. This is a fungus, pneumocystis, that's latent in the lungs. It can start growing once the CD4 count's under 200. Toxoplasma is a parasite that's latent in the brains. For those of you who, when you were kids and played in sandboxes and ate the cat poop, about 30% of you did that if you wanted to know about it. And that can cause brain abscesses. And then I mentioned cytomegalovirus, CMV. So the goal, obviously, this is the natural history I'm reviewing and repeating there. You get in sex here, huge burst of virus, you've got the mono-like symptom, sickness, you know, fever, sore throat, headache, and a rash. Immune system kicks in, virus drops down, you develop antibodies to the virus so you can test for the cheap antibody test and discover you're infected. Eventually, CD4 cells drop. You get, once the CD4 count drops lower, you start reactivating all those bad infections that are latent in you and you get sick. How do we diagnose HIV? We put antigens in plastic wells. We then put on the patient's blood, and if they have antibodies to it, it'll stick to the antigen, all right? And then we put in antibodies against human antibodies that have a marker to it. In this case, it is a color. So these are wells that have patients' blood that was thrown in them. That each well has pieces of HIV antigen in the base of it. The antibodies stick to it, and then we put in the antibodies that are against human antibodies, so that if there's antibodies sticking there on the HIV, it'll make this red color, and that's how we know someone's infected. We also confirm it in the United States using what's called a Western blot. That spreads all those HIV antigens out by charge and weight, and you look for these black bands here, or sometimes they're in color, and you can see this patient had an antibody against GP120 and against GP41, these other HIV proteins, and we can confirm that they're infected. HIV is in semen, we know. This is work done at UNC. And when you treat the patients, the amount of HIV goes down in the semen. It's also in cervical washings, and when you treat it, the amount of virus goes down in cervical washings. So without treatment or anything like that, the CD4 count drops, the amount of virus goes in, and the patients die. With early treatment, you suppress the virus, you preserve the immune system, and the patients can be asymptomatic and live many, many, many years. And this is an example from a very, very early study that we did here in Chapel Hill, and it showed what happens to the mortality even when we weren't giving triple drug therapy. So in this case, we started out with 1 million copies of the virus, this patient here, that when we dropped, and he had 100% risk of death by the end of 3 years with that much virus in the blood. When we dropped the amount of RNA in a patient with that high by 1 log, so that's still down to 100,000 copies from 1 million copies, it decreased the mortality by 2/3. If you drop it down 2 logs, which is still 10,000 copies in the blood, it dropped down to 13%. So dramatic effect by decreasing the viral load on mortality. And so what happened, so remember I started taking care of these first patients in 1980 there. It ended up from that period of time where it was the leading cause of death on American men and women of this age group, 25 to 44. When we started treatment, when we discovered that triple drug therapy could suppress the virus completely, all of a sudden it dropped off and people had stopped dying. That's it.